Good day. This is the Professor, CBMD PhD. And today is Wednesday, May 17th, 2023. It is 3:38 and 8 seconds, a.m. Eastern Daylight Time. And today's research, slow and engaging. So let's hop to it here. The feeling of hunger itself may slow aging in flies. Date May 11, 2023. Source, University of Michigan. While it has long been understood that limiting the amount of food eaten can promote healthy aging in a wide range of animals, including humans, a new study has revealed that the feeling of hunger itself can be enough to slow aging. As previous research has demonstrated that even the taste and smell of food can reverse the beneficial life-extending effects of diet restriction, even without its consumption. Research investigators and their colleagues set out to examine whether changes in the brain that prompt the drive to seek food could be behind longer life. They sought to divorce the life-extending effects of diet restriction from all the nutritional manipulations of the diet that researchers had worked on for many years to say they are not required. The perception of not enough food is sufficient. And to do this, they induced hunger in flies in several ways. The first was to alter the amount of branched chain amino acids in the test snack food, and later allow the flies to freely feed on a buffet of yeast or sugar food. The flies fed the low branched chain amino acid snack consumed more yeast than sugar in the buffet than did the flies fed the high branched chain amino acid snack. This kind of preference for yeast over sugar is one indicator of need-based hunger. The researchers noted that this behavior wasn't due to the calorie content of low-branched chain amino acid snack. In fact, these flies consumed more food and more total calories when flies ate a low-branched chain amino acid diet more light. They also lived significantly longer than flies fed high branched chain amino acid diets. And to look at hunger, apart from dietary composition, they used a technique activating neurons associated with the hunger drive in flies using exposure to red light, using a technique called optogenetics. As these flies consumed twice as much food as did flies who were not exposed to the light stimulus, the red light activated flies also lived significantly longer than flies used as a control. The scientific researchers say they've created a type of insatiable hunger in flies, and by doing so, the flies live longer. What is more, the team was able to map the molecular mechanics of hunger to changes in the epigenome of the neurons involved, and to identify that neurons responded to the presence or absence of a specific amino acid in the diet. And these changes can affect how much of specific genes are expressed in the brains of flies and consequently their feeding behavior and aging. The scientific investigators know that caution should be used before applying the findings to people, but there's every reason to expect that the mechanisms discovered are likely to modulate hunger drives in other species. The next plan to examine how the drive to eat or pleasure present in both flies and people may also be linked to lifespan. And this is the Professor of Microbiology, Bioinformatics, Pathophysiology, and leader of research. This video was created for educational purposes only. Hope you enjoyed listening. Until next time, have an awesome day.